Okay, so today we're going to be talking about photosynthesis, and essentially, photosynthesis utilizes the energy from sunlight that, in turn, transfers this energy into chemical energy through sugars, as we will later see. It is important to learn about photosynthesis, as it is a key process for understanding how animals like us or other species obtain nutrients and energy through eating fruits like these peaches or vegetables after photosynthesis occurs.
In order for photosynthesis to take place, 3 products are needed: water, carbon dioxide, and light energy from the sun. These 3 products are usually absorbed by producers in order to generate their own energy source needed to maintain life. The resulting products are glucose, which is used as energy, and oxygen gas, which is released into the atmosphere as a waste product.
So here we are going to look at the action and absorption spectrums. With these 2 spectrums, we can view how intensity of the sunlight affects the ATP production rates within the plant during photosynthesis. The action spectrum specifically introduces the rate of photosynthesis for the different colored wavelengths of light and the percentage of the maximum rate of photosynthesis that occurs while a specific color of light enters the plant. This introduces the absorption spectrum, looking at how different pigments of the wavelengths of light are absorbed. So within plants, we know that reds and blues, or violets, are highly absorbed, while green and yellow pigments are often reflected rather than absorbed, which is why we see most plants as green or yellow.
Photosynthesis takes place inside the plant cells. More specifically, they occur within structures called chloroplasts. Within the chloroplast, one can find small disc-like structures called thylakoids. These are surrounded by a fluid called the stroma and connected by a structure called the lamella. 2 reactions take place within the chloroplasts. These 2 reactions are light-dependent reactions and the Calvin cycle. Light-dependent reactions take place within the thylakoid membrane. Here, photosystems containing chlorophyll absorb the light energy from the sun and convert it to chemical energy. Electrons are excited within the photosystems and are transported out. Because these electrons have been transported outside of photosystems too, they are replaced by electrons generated by the splitting of water into oxygen and hydrogen ions using light energy. This is called photolysis. Hydrogen ions are then pumped into the thylakoid using the energy generated by the electrons as they pass along the electron transport chain. This creates a concentration gradient which is then used to power a protein called ATP synthase. The phosphorylations of ADP to ATP take place here. After the electrons are re-energized by photosystems 1, they are used to reduce NADP+ into NADPH. The ATP and NADPH produced in the light-dependent reaction are then released into the stroma to be later used in the Calvin cycle.
Essentially, the Calvin cycle consists of the carbon dioxide being reduced to create glyceraldehyde 3-phosphate. The first stage takes the carbon dioxide molecules and connects them with ribulose 1,5-biophosphate, or also known as RUBP, which then splits from 6 to 3 carbon molecules, these molecules being PGA, which is phosphoglycerate. The 2 NADPH and ATP now play their part as electrons are used to cause reactions that will further reduce the carbon atoms.
The regeneration of ribulose 1,5-biphosphate occurs in the final step. 5 molecules of glyceraldehyde 3-phosphate are used every 3 cycles in order to produce 3 molecules of ribulose 1,5-phosphate. Glucose is made from the remaining glyceraldehyde 3-phosphate. Fatty acids or glycerol can also be produced. 2 molecules of glyceraldehyde 3-phosphate are needed to make 1 molecule of glucose phosphate. For this reason, the Calvin cycle must run a total of 6 times in order to make a single molecule of glucose.